into endoparasites. And we will discuss all these aspects, not only from the view of the host, but mainly on the interest what the parasite protects them from being poisoned by the host. Or are these parasites resistant to that or not? Are these or some animals also acquiring resistance against the action of such a paradise? So this is an ugly picture. This is a dying or even almost dead toad, which had been attacked by a small fly. This small fly, like blowfly, which are, well, you know, they are very common. If you have a cake here or something, there will be in 5 minutes be around you. But these are specialized for toads only. They attack toads only. And if toads are not available, eventually they take a frog. But in most cases, they search, they're not very abundant, but they search for a toad and place their eggs on the head and on the shoulder of the toad. And the hatching larvae then enter the nostrils of the toad, enter the body, and, well, consume the toad from the inside out. So that is the monster in the body. And you see here that it's a dying toad, or it's a dead toad already. Nothing left after that since the skeleton of the toad. In some areas of Germany, there had been a really good investigation how often that occurs. In some populations, only sometimes 70% of the toads are eradicated by just these small specialized flies. Well, at the end of the horror story, so you see most of them had been consumed in the inner part of the body, but you see also other maggots are around. These are not these maggots from that fly, Buffonivora, but from other of the other Lucilia genus, which just feed on carcass. They attracted by carcass only, or dead animals, including humans, of course. The science, the forensic science people have a great interest in that. But that is an example that both species are closely living together, but they are exposed to the same part of toxins. These are bufadenial, I have a problem sometimes, bufadenialites, very complex molecules, but very close related to digitoxin and also these plant cardiotoxic cardenolites. They secreted the toads mainly here in the parotoid glands. And if you take a sample for that and do HPLC, you can find a high concentration of them here in this fraction. And when we use them, we pick the larvae and make the same extraction with the larvae, and you find these various cardenolites, at least 20 different molecules sometimes, in the larvae. And the main toxin is bufotoxin. And this is the main peak here. And you see, they are present in the larvae. The larvae are not dying, by the way. And they reach even the pupation, and from the pupa, the new fly hatches. The fly is entirely free of any of these toxins. So something must be special with with these flies, that they tolerate the high toxicity of the bufadenialites. What is that happened? We know already that the main target of bufadenialites, and the same also for cardioactive glycosides are the sodium, potassium ATPase. And we also know that this enzyme has an alpha and a beta subunit, and the main binding site of bufadenialites as well as cardioglycosides are the alpha site. And there are two places of two sides of this sequence number 111 and 122. This is glutamine and asparagine, which are the binding sites of the sodium of these toxins. And you see, when we compare that, Drosophila melanogaster is sensitive to it. You can kill them with these low concentrations of the toxin. But also, you see here, Lucilia buffonivora, this toad fly, has the same structure. And Lucilia vigilia, the maggots feed on carcass, had the same unchanged structure. They must be sensitive. This enzyme must be sensitive to these toxins. But how they survive? What's the trick to avoid intoxication? We know, speculate more or less, that they have a very efficient excretion mechanism. They can store it in the gut, quite a concentration of these toxins, but the toxins do not permeate, obviously, the membrane of the gut. So they excrete it and get rid of it, and finally the fly is free of and even the pupa has a very tiny concentrations only of these toxins, but the fly is entirely free. So they get rid of these toxins separately from the rest of what they consuming is the main answer. Here we have what I presented first, the so-called African queen, wonderful name, by the way, because this is a relatively of the monarch butterfly in North America, and they lived in Africa. And I have been many times in Namibia, and if you come into to Namibia in springtime, millions of them are around. They're flying everywhere, and I'm so keen to collect the small, slightly growing caterpillars and reared them at home. 30, 40 I took, and to my disappointment, 90% are parasitized. So you see here the parasites from the living caterpillar come out. And after a while, they form pupation. They make the pupation. And in that caterpillar, for instance, 50 pupae are present. And it's only placed by a tiny wasp, not larger than, you see, the millimeters only, no, 3 or 4 millimeters body size, and injects tiny eggs, up to 50 of them, and kill a number, or a huge number of these caterpillars. And the next question is, how these caterpillars, not the caterpillars are killed, of course, to my disappointment, but how these maggots or the larvae, how they survive in an environment of a body which is full of plant products, material, full of these, what I show you here. No, come on. Oh yeah, it's the same structure. I didn't place it here. The same structure like the buffodanilides. The annulites are the glycoside, the cardiac glycosides, which are in the plant. And when we, and I haven't done this personally, but my dear friend Susanne Dobler from the University of Hamburg was kind enough to analyze the same situation like here, the sodium potassium ATPase, and where the binding site exists for these maggots or for the larvae, for the toxin in the larvae. And what we see here, it is well known, of course, Drosophila melanogaster is highly sensitive to the toxin, but Danaus plexipus, this is a monarch of the United States, has a replacement of asparagine by histidine, which makes this enzyme resistant, or the toxin cannot bind. And interestingly enough, the wasp, Draconid wasp, has also a replacement, but not on this situation here, but in the beginning on 111, also replacement with histidine. Histidine in general prevents binding of the toxin. This is a very interesting finding. So you can say this is a good explanation how the parasite adapted to the target organ that the toxin cannot bind to that point. This is my, 2 minutes. Yeah, this is my favorite place in Pennsylvania, a pond where I'm studying since 12 years now, continuously, the presence of these two newts. Red spotted newts, Nophthalmos and viridescens, which contain high concentrations of tetrodotoxin, just to tell you. But over the years, I was surprised to find that more and more in this particular pond, the newts are parasite, contain these modifications with big changes in the skin. And when you do microscopy, you can isolate a wonderful fluke, not larger than 2 millimeters, but it's like here in this one. But it must have contact to the high concentration of tetrodotoxin. When you dissect these newts, you see this is a liver completely filled with what we see here, with cysts. And these cysts accumulate to completely destroying the liver, including also under the skin. What is particularly interesting is here we have the secretory cells with high concentration of tetrodotoxin, but below that are cysts which have a big wall, which obviously prevents the penetration of tetrodotoxin into these cysts. And they developed, the fluke developed to a certain stage. Interestingly, when we discuss, some are contain parasites, others do not contain. If we estimate the tetrodotoxin concentration that's here and an analog, there's no difference between, or no significant difference between the two. So it doesn't play a role whether the newt is containing tetrodotoxin or not. Interestingly, this is the life cycle of such parasite. The parasite is excreted as eggs from a bird, introduced by a bird, and larvae hatch, enter another host, this is a snail, and the snail, in the snail, develop small free-swimming cercaria, which enter and fish, or even our hosts newt, and form these, what I showed you, these capsules or these cysts. And all those had to be eaten by a bird, because this is the final host where they reach a stage good enough for sexual reproduction. Bad for the parasite here is the newt. The newt is not eaten by a bird because it's too toxic. The birds avoid to eat them. So this is dead end for the parasite. But in fish, for instance, then the cycle starts again. This is an interesting phenomenon which always tell you how complicated the interaction sometimes is, and how necessary it is to change from one host to another and still being resistant to toxic activity. So what is the conclusion? So the poisonous animals seem to be well protected from the attack of predators. We are not spared from the infestation by parasites. Despite the concentration of the toxins, they are really defenseless against parasites. You cannot do anything. You live in an area where they are present or not. If the fly is around, okay, then it's the end of the toad. But they cannot do anything against it. The toxin doesn't help to protect them. And parasites develop interesting various mechanisms. A part of them we understand now. But there must be huge numbers of other resistant mechanisms which prevent the parasites to be inhibited to enter this host, or to come to an end with the their life cycle. This includes, for instance, what we now already is efficient excretion, get rid of the toxins, or structural adaptation to the target of the toxin involved. And finally, I want to thank my former collaborators in the Institute of Legal Medicine in France. We still tolerate my presence sometimes in the Institute, but also when I approach them, can you help me in this and this situation to do some analysis? They do it and are quite helpful. And without them, I wouldn't be here. Thank you very much for your attention.